Pharmapal.
Immunotherapeutic targeting of mesothelin-positive pediatric AML using bispecific T-cell engaging antibodies. Hey everyone! Welcome back to our channel. Today, we're diving deep into an exciting area of cancer research, specifically focusing on pediatric acute myeloid leukemia, or AML for short. We'll explore how scientists develop innovative treatments using bispecific antibodies, or BiSABs, to target this challenging disease. So, grab a snack, get comfy, and get started. Acute myeloid leukemia is a type of cancer that affects the blood and bone marrow. It's particularly tough on children, and researchers are constantly looking for new ways to improve treatment outcomes. One promising approach involves using bispecific antibodies, special proteins designed to engage the body's immune system to fight cancer cells more effectively. So, what exactly are bispecific antibodies? Well, they are engineered to bind to two different targets at the same time. In our case, these BiSABs are designed to target mesothelin, a protein commonly found on the surface of some cancer cells, and CD3, a component of T cells, which are crucial players in our immune response. The researchers in this study combined sequences from two well-known antibodies to create their own BiSABs that target both mesothelin and CD3. They called these Mesothelin CD3L2K and Mesothelin CD3AMG. The goal? To redirect T cells to attack and kill mesothelin-positive AML cells. To measure the effectiveness of these BiSABs, the researchers conducted various experiments. They calculated the percentage of human cells in their models using a specific formula, which helps to quantify how much of the human immune response is involved in fighting leukemia. Now, let's talk about the experimental setup. The team used a pediatric AML cell line called MV4-11, which was modified to express high levels of mesothelin. They co-cultured these modified cells with T cells from healthy donors to see how well the BiSABs could induce T cell-mediated cytotoxicity, basically, how effectively the T cells could kill the cancer cells. The results were promising. Both BiSABs showed a significant ability to kill the mesothelin-positive AML cells. Interestingly, Mesothelin CD3AMG was more effective than its counterpart. This difference in effectiveness was linked to the varying affinities of the antibodies for their targets. But it's not just about killing the cancer cells. The researchers also measured cytokine release, signaling molecules that help coordinate the immune response. They found that the BiSABs triggered a strong cytokine release, indicating that the T cells were not just killing the cancer cells, but also activating a broader immune response. The scientists tested these BiSABs in live animal models to take things a step further. They injected the modified AML cells into mice and monitored how the treatments affected leukemia progression. They found that the Mesothelin targeting BiSABs significantly reduced the percentage of AML cells in the bone marrow of treated mice compared to controls. In terms of survival, the data was even more encouraging. The mice that received the Mesothelin targeting treatments showed improved survival rates compared to those that did not. This is a crucial indicator that these BiSABs could potentially translate into better outcomes for children with AML. To wrap things up, this study highlights the potential of bispecific antibodies as a novel immunotherapy for pediatric AML. These BiSABs could offer a new lifeline for young patients battling this aggressive disease by targeting mesothelin and engaging T cells. Now, AML is a type of cancer that affects the blood and bone marrow, and it's particularly challenging in children. But researchers are making strides to improve outcomes, and that's what we're here to talk about. In this study, scientists have engineered bispecific antibodies, or BiSABs, that target a protein called mesothelin, which is often found on the surface of AML cells. What exactly are bispecific antibodies? Well, they're special proteins designed to engage two different targets at once. In this case, one end of the antibody binds to mesothelin on the cancer cells, while the other end engages T cells, which are a type of immune cell that helps fight off infections and diseases, including cancer. By connecting these two components, the BiSABs can redirect T cells to attack the leukemia cells more effectively. This innovative approach aims to harness the body's own immune system to combat the cancer, which is a promising strategy in cancer therapy. The researchers conducted a series of experiments to evaluate how well these BispCiffs work, both in the lab and mouse leukemia models. They wanted to see if these antibodies could target the cancer cells and activate the T cells to effectively eliminate leukemia. So, stick around as we explore the details of this exciting research and what it could mean for the future of treating pediatric AML. Now, AML is a type of cancer that affects the blood and bone marrow, and it's particularly challenging in children. But researchers are making strides to improve outcomes, and that's what we're here to talk about. In this study, scientists have engineered bispecific antibodies, or BispCiffs, that target a protein called mesothelin, which is often found on the surface of AML cells. You might be wondering, what exactly are bispecific antibodies? Well, they're special proteins designed to engage two different targets at once. In this case, one end of the antibody binds to mesothelin on the cancer cells, while the other end engages T cells, which are a type of immune cell that helps fight off infections and diseases, including cancer. By connecting these two components, the BispCiffs can redirect T cells to attack the leukemia cells more effectively. This innovative approach aims to harness the body's own immune system to combat cancer, which is a promising strategy in cancer therapy. The researchers conducted a series of experiments to evaluate how well these BispCiffs work, both in the lab and mouse leukemia models. They wanted to see if these antibodies could target the cancer cells and activate the T cells to eliminate the leukemia effectively. Next, let's talk about the cell lines used in this study. The researchers worked with a couple of key players, MV4-11, a pediatric AML cell line, and OVCR3, an ovarian carcinoma cell line. They also created a modified version of MV4-11, called MV4-11-MSLN, by introducing the human mesothelin gene into the cells using lentiviral transduction. This technique allows the researchers to insert new genetic material into cells, essentially giving them new abilities, in this case, to express mesothelin on their surface. Now, how did they measure the expression of mesothelin? They used a technique called flow cytometry. This method involves tagging the cells with fluorescent antibodies that bind specifically to mesothelin, and then passing the cells through a laser beam. The fluorescence is measured, allowing scientists to quantify how many mesothelin antibodies are bound to each cell. Pretty cool, right? Next, let's discuss the bispecific antibodies, or BispCiffs. These are engineered proteins that can bind to two different targets at once, in this case, mesothelin and CD3, a component of T cell receptors. The researchers created these BispCiffs by combining sequences from existing antibodies, specifically amituximab for mesothelin and blinatumomab or AMG 330 for CD3. They expressed these antibodies in HEK293 freestyle cells, a common choice for producing proteins in the lab. Once the BispCiffs were produced, they needed to be purified. This was done using affinity chromatography, which separates proteins based on their specific interactions, followed by size exclusion chromatography to ensure the proteins were the right size and pure. The final products were then ready for testing. Now, let's move on to the in vitro assays. The researchers co-cultured the MV4-11-MSLN cells with freshly isolated T cells from healthy donors. They used a specific ratio of T cells to target cells, called the effector-to-target ratio, set at 5:1. This means for every 5 T cells, there was one target cell. They added the BispCiffs at different concentrations to see how effective they were at killing the AML cells. To quantify the effectiveness of these treatments, they calculated the percentage of cytotoxicity using a formula that compares the number of live cells with and without the drug. They also measured cytokine release, which indicates how active the T cells are during the immune response. Finally, for the in vivo experiments, the researchers injected the modified MV4-11-MSLN cells into mice to create xenograft models. They then treated these mice with the BispCiffs and monitored their health, looking for signs of leukemia progression. This involved drawing blood and analyzing it to see how many human AML cells were present. Now, results from a recent study on bispecific antibodies, or BispCiffs, target a specific protein called mesothelin, or MSLN, in pediatric acute myeloid leukemia, also known as AML. So, what are bispecific antibodies? They are engineered proteins that can simultaneously bind to two different targets. In this case, the researchers designed BispCiFs to target both Mesothelin, often overexpressed in AML cells, and CD3, a part of the T cell receptor that helps activate T cells to attack cancer cells. Now, let's get into the results. The study showed that the Mesothelin targeting BispCiFs were super effective in promoting T cell activation and killing AML cells in laboratory settings. They used a specific AML cell line called MV4-11, which they modified to express high levels of Mesothelin. When they co-cultured these modified cells with T cells, the BispCiFs induced significant T cell-mediated cytotoxicity, meaning the T cells were able to effectively kill the cancer cells. Now, let's talk numbers. The researchers measured how well these BispCiFs worked by calculating the effective concentration, or EC50, which is the concentration needed to achieve 50% of the maximum effect. For one of the BispCiFs, named CD3-AMG, the EC50 was around 0.43 nanograms per milliliter, which indicates a strong potency. In contrast, the other BispCiF, CD3L2K, had a much higher EC50 of 4.82 nanograms per milliliter, making it less effective in comparison. But that's not all. The researchers also conducted in vivo experiments using mice models. They injected these mice with AML cells that expressed Mesothelin and then treated them with the BispCiFs. The results were promising. Mice treated with the Mesothelin targeting BispCiFs showed significantly reduced levels of AML cells in their bone marrow compared to control groups that did not receive the treatment. One of the key findings was that the BispCiFs not only reduced the leukemia burden but also improved the overall survival of the mice. In fact, the study highlighted that the Mesothelin targeting BispCiFs prevented the development of extramedullary disease, which is when leukemia spreads outside the bone marrow, a common and serious issue in pediatric patients. Next is the findings from Mesothelin, especially in the context of pediatric acute myeloid leukemia, or AML. Now, let's break this down. First off, what are bispecific antibodies? Well, these are engineered proteins that can bind to two different targets at the same time. In this study, the researchers focused on creating BispCiFs that could engage T cells, our immune system's warriors, while also targeting Mesothelin on leukemia cells. This dual action is crucial because it helps direct T cells to the cancer cells, enhancing the immune response against the leukemia. The study highlighted that these Mesothelin targeting BispCiFs were particularly effective in promoting T cell activation and reducing the leukemia burden in mouse models. This is significant because pediatric AML can be quite aggressive and challenging to treat. The researchers used two different types of BispCiFs, one based on a well-known antibody called blinatumomab and another based on AMG 330. They found that the AMG 330-based BispCiF was more efficient in inducing T cell-mediated cytotoxicity and producing important signaling molecules called cytokines. So, why does this matter? Well, the success of blinatumomab in treating acute lymphoblastic leukemia has opened doors for similar strategies in AML. The findings suggest that targeting Mesothelin could be a promising approach for immunotherapy in pediatric AML, potentially transforming treatment options for these young patients. Now, let's talk about the models used in the study. The researchers created a special mouse model that mimics the human disease by implanting leukemia cells. They carefully monitored the mice's health and leukemia progression through blood tests and post-mortem analysis. This allowed them to evaluate how well the BispCiFs work to reduce leukemia cells in the bone marrow, a critical measure of treatment efficacy. Interestingly, they noted that the Mesothelin targeting BispCiFs not only cleared the bone marrow but also helped prevent the development of extramedullary disease, tumors that can form outside the bone marrow. This is particularly important because extramedullary disease can complicate treatment and worsen outcomes in pediatric patients. The researchers also compared the two BispCiFs in terms of their effectiveness. They found that the AMG 330-based BispCiF showed superior performance in both in vitro and in vivo settings. This could be due to its higher affinity for the CD3 protein on T cells, which plays a crucial role in T cell activation. In conclusion, this study provides compelling evidence for the potential of Mesothelin targeting BispecificTCells as a novel immunotherapy strategy for pediatric AML. It opens up new avenues for research and could lead to more effective treatments for children battling this challenging disease. Thanks for tuning in. If you found this video informative, don't forget to like, subscribe, and hit that notification bell for more updates on the latest in cancer research. See you next time.
Pharmapal.